Welcome to the first lesson of Medical Point. This is also the first topic of our cell biology unit. Today we will be covering if viruses are alive or not, the structure of viruses, and their classification. Feel free to pause the video anytime and rewatch. Just in case you want to follow along, you can find the digital notes that correspond with this lesson on our website. The website is linked in our Instagram bio if you need it. To begin with, what is a virus? A virus is a submicroscopic infectious agent that replicates only inside the living cells of an organism. Viruses infect all types of life forms, from animals to plants to microorganisms, including bacteria and archaea. So, most of the time, it is inside of a living organism. But does that mean it's also living? For example, our stomach is inside us and humans are in fact living, but our stomach is not. I know stomachs and viruses are two completely different things, but is it the same case? Let's find out. In order to find out if a virus is living, we can use the 7 characteristics of life. If something has all 7 characteristics, it can be labeled as a living organism. The first characteristic is being able to maintain homeostasis. Just for a reminder, homeostasis is a process of maintaining a stable internal environment. Viruses do not pass this requirement since viruses do not have nuclei, organelles, or cytoplasm like other cells do. They do not have any way to monitor and create change in their internal environment, so they fail this test. The second characteristic of life is having a cellular organization. Viruses do, in fact, pass this test. Viruses have genes made up of nucleic acids and a capsid made of capsomeres, which are smaller subunits. This correlates with the idea that smaller building blocks come together to make a larger product. This picture depicts an example of a cellular organization in humans, but we'll look into the structure of a virus later. The third characteristic of life is being able to reproduce. It is still unsure whether viruses pass this requirement. Even though viruses definitely multiply, they can only use the host cell in order to do it. This is because viruses do not have the organelles, nuclei, or ribosomes that are necessary in order to copy their genetic material or duplicate. But when these viruses enter living cells, they are able to use the host cell's equipment to replicate and copy genetic information. So all in all, they can only reproduce in host cells and not by themselves. We will go over the reproduction cycles for viruses in the next video, so be on the lookout for that. The fourth characteristic of life is being able to grow and develop. Viruses do not pass this requirement. This is because viruses manipulate host cells into helping it replicate and build new viruses. This means that each virion is created in its fully formed state and doesn't grow throughout its existence. The fifth characteristic is metabolism. It's still unsure whether viruses pass this requirement because creating new virions from building nucleic acids and putting capsid together takes a lot of energy. But all of this energy comes from the host cell and doesn't come from the virus itself. The sixth characteristic is being able to respond to stimuli. It is still unknown whether the virus passed this requirement because responding to stimuli is defined as an almost immediate reaction to some change in the environment. There's still not any research done to find out if viruses respond to anything yet. So this will be unknown until they do so. The seventh characteristic is being able to adapt to the environment. Viruses do pass this requirement. Adaptations happen through mutations that have advantages on species. Viruses definitely adapt to the environment. An example is their switching between the lytic and lysogenic cycle. The lytic and lysogenic cycle is the two cycles a virus use in order to replicate. The virus can only use one. The lytic cycle is when a virus actively replicates in a host cell, and the lysogenic cycle is when a viral DNA is incorporated into the host cell's DNA and either replicates or remains dormant. A virus that is in the lytic phase can switch to the lysogenic if the host does not have enough energy or supplies to support the virus to actively replicate. Sometimes viruses can end up switching back to the lytic again when conditions are correct. This signifies that viruses' ability to adapt to the host's condition. In summary, viruses are definitely not dead as they contain some of the different characteristics, but many biologists say that viruses are not alive either. This is because they don't follow any of the characteristics except for the ability to replicate and adapt to the environment. So it is still unknown whether they are actually alive or dead, but they just may happen to be in between both categories. Next, we will be talking about the structure of a virion and how to classify a virus. But first, we need to understand more about the virus and learn what a virion is. The main purpose of a virus is to deliver its genome to the host cell to allow its expression which is transcription and translation by the host cell. When infected, a host cell is forced to rapidly produce thousands of identical copies of the original virus. When not inside an infected cell or in the process of infecting a cell, viruses exist in the form of independent particles or virions. Virions are functional viruses with the capacity to infect a living tissue. Basically, if a virus is extracellular, which means it's found outside of the cell, it is a virion. Virions consist of one, genetic material, two, a protein code called the capsid, and in some cases, three, an outside envelope of lipids. The genetic material can be long molecules of DNA or RNA. They encode the structure of the proteins by which a virus acts. The capsid functions as a cell to protect the viral genome. During infection, the proteins of capsid attaches the virion to specific receptors exposed on the prospective host cell. Capsid proteins are encoded for by the virus genome. Some virus families have an additional covering called the envelope, which is usually derived from part of modified host cell membranes. Viral envelopes consist of lipid bilayer that closely surrounds a shell of virus encoded membrane associated proteins. The exterior of a bilayer is studded with virus coated glycosylated membrane proteins. If you need to see the structure of a virus picture up close, here it is. You can see the virus has all three parts that we discussed and the membrane proteins, which are labeled as an envelope protein here in this picture. On to the classification of a virus. You can classify a virus by its shape, nucleic acid, and the type of host. So generally, a virus is really, really small. Bacteria are 100 times larger than a virus. So now you should be able to get a perspective since bacteria are already microscopic. Some viruses are smaller than others, so that should narrow it down a little bit more. Next, you can use its shape. The capsid shell builds the shape of the virus. Remember that the capsid is the protein coat that protects the viral genome. The different shapes in a virus are polyhedral, complex, helical, and spherical. We'll see this really soon. Moreover, a virus can either have a single-stranded DNA or a single-stranded RNA. It can also have a double-stranded DNA or a double-stranded RNA. It is unique to a virus to have a single-stranded DNA and a double-stranded RNA, but it can only be one of the four. So nucleic acid helps distinguish viruses from one another. This genetic information needs to be packaged so it is stored inside the protein code. So a virus is created using a capsid and nucleic acid, which is called a nucleocapsid, which is why the previous had nucleocapsid instead of just capsid. To narrow it down even further, viruses can be classified by the type of host they infect. This includes animal, bacteria, and human and plant cells. Here are the four types of shapes that a virus can be, with corresponding examples. See if you can take these 6 viruses and group them into the shape categories without going back to the other slides. You can go ahead and pause the video for time to categorize. Moving on. A virus is an obligate intracellular parasite, which means it hacks into other things to survive. So, how does the virus even enter into the cell? Bacteriophages have a complex shape in which their tails attach itself to the host body. So the sheath can inject the bacteria viruses using receptors to their advantage in order for them to sneak in into the cell. If the virus has an envelope, it could enter by fusing with the cell membrane, aka direct fusion. We'll look more into this into the next video. I hope this lesson was helpful to study for the MCAT or just for regular science class. Make sure to subscribe to the YouTube channel and thank you so much for watching.